It is my great pleasure to introduce you our today's speaker, Nora Bengoa. Nora completed her PhD in 2014 at the University of the Basque Country, where she studied the role of Wnt signaling during early neuronal differentiation in human stem cell models. She then joined the Wade Martins lab at Oxford University in 2015 for her postdoctoral training, focused on the role of alpha-synuclein aggregation and glial activation in Parkinson's disease. At Oxford University, later on 2018, she became a research fellow. And since last year, she established her own research laboratory at the Achucarro Basque Center for Neuroscience. Her research interests are focused on alpha-synuclein aggregation and neuroinflammation in neurodegenerative processes, specifically in Parkinson's and Alzheimer's disease. So I would like to thank Nora for her time and her willingness to share with us her work. And Nora, welcome to this webinar series. Welcome to CIBERNED, and the floor is yours. So please share your screen whenever you're ready. Thank you.
Thank you very much for that kind introduction, and also for the invitation to speak. It's been a few months almost a year since I established my lab, so I'm fairly new to the research community, but I found it very welcoming so far. So today I'm going to tell you a bit about the story that we recently published in Nature Comms, in vitro and in vivo protection of neurons by the molecular tweezer CLR01. But I also wanted to give you a flavor of a different story. So I'm going to talk firstly about this particular project, and then about an ongoing one that Michael J. Fox, the Michael J. Fox Foundation, has actually given us further funds to continue. And I'll just start with a very brief introduction, and I don't think that I need to actually explain to this crowd why we actually go to the lab each day, but it is because neurodegeneration is the biggest socioeconomical burden on society today. We know we live in an aging population. We know that the most overrepresented diseases of neurodegeneration are firstly Alzheimer's, and then Parkinson's, and millions of people estimated at 50 currently live with dementia worldwide. And this figure is predicted to triple by 2050. So we have a really phenomenal problem that we're not really addressing, in my view, perhaps properly. And because today I'll only talk about Parkinson's disease, I'll use Parkinson's as an example of why I think this is an unmet need. So in 1817, James Parkinson writes his essay on the shaking palsy, where he describes the cardinal symptoms of Parkinson's disease. And we've just celebrated a few years ago the 200th anniversary. But it isn't until 1912, when Frederick Lewy describes Lewy bodies, that to this day remain the only unequivocal way of diagnosing postmortem Parkinson's disease. And it is in 57 when Consorcio shows that dopamine is a neurotransmitter, and shortly after Coetzee shows the efficacy of levodopa, which with modifications such as carbidopa, etc., has remained the main therapeutic agent that we have to this day, which is, we have to remind ourselves, not disease modifying. And finally, in 97, Maria Grazia Spilantini shows that there's alpha-synuclein in Lewy bodies. And perhaps it would be worth, we come and go about whether we want to add DBS as a therapeutic for Parkinson's disease. And because it touches quite few patients worldwide, it doesn't meet it. But regardless of whether it meets or it doesn't, this shows you that we're missing more crucial milestones, so accelerating therapies to patients. We're really not advancing at the pace that other conditions, such as, for example, you think about cancer and how triple negative breast cancer has advanced, or how we're getting new therapies for other diseases. Neurodegeneration suffers from a block, and there's many things that actually makes sense for this block. The brain is a very difficult organ to study. The obtaining of biopsies is very complicated compared to other diseases such as cancer, etc. But it leaves us with this clinical landscape, and this is the CPT-inspired annual review that either has just come out or the new one will come out this month or next. With the previous year in review, what the PD therapeutics looks like in this case, the previous one, 2021. And what we can see on the top part of the graph is where we are in clinical phase 1, 2, and 3 and disease-modifying therapies, and under the bar is symptomatic relief. And it's just striking to see that there's far more efforts being put into symptomatic relief. And that's the problem. We haven't hit the crux of what it is that is disease modifying. And it is a problem to have such a empty space here at phase 3, because it means that if we don't really have candidates for phase 3, we're not really going to meet that deadline soon. The one thing that really personally I find good is the number of colors that we have on the top part of this graph. So there are abundant, because there are many scientists claiming that we only focus on amyloid and we only focus on antibody therapies, anti-aggregates. And I don't really agree that that's the case. We can see here that there's a range of things that we're trying to target at the clinical level, and that both industry and academia, I think, are taken by many different hypotheses. And I think that's encouraging to have to look at different flavors, since we don't know what the crux of the matter is. In the lab, we have specialized in the detection and the study of early aggregates. And we believe that through whether loss of function or gain of function, these interfere with disease. We don't believe they're the sole drivers of disease, but we do think that alpha-synuclein early aggregates such as oligomers or short fibrils play a role in disease. And we think that this has not only been shown by us, but in this review, we looked at the literature, and many groups have shown that oligomers can cause autophagic and lysosomal block, thereby leading into vicious cycles whereby there's more aggregates, so there's more block, so there's more aggregates, etc. There's ER stress and dysfunction caused by oligomeric accumulation intracellularly, as well as proteasomal dysfunction, which ends up clogging up all the waste management system of the cell. Mitochondria have been shown to fragment. We know that complex 1 dysfunction is associated to oligomeric deposition, and that these can punch holes in membranes, leading to membrane damage, both of the plasma membrane and in the mitochondria. And at the level of the synapse, we know that they can interfere with vesicle traffic and synaptic formation. And outside of the cell, there's a missing astrocyte, but we also know that they can interfere with other cells in the brain and thereby cause aberrant inflammatory responses. And so we think that they are quite important, a quite important part of the disease that we really need to address. But we also acknowledge that they're very difficult to work with. When you make them in the recombinant flavor, they're very dynamic species. It's very difficult to get reproducible preps. And at the detection level, when I started originally in the lab, we were really not taken by the specificity of a lot of the antibodies that claimed that they were oligomeric specific in the market. And so we decided to invest a few years of several postdocs' lives, including mine, to the development and optimization of the alpha-synuclein proximity ligation assay. And so this assay, or proximity ligation, generally uses an antibody targeting one protein and a second antibody targeting a different protein. You label one with a plus probe, one with a minus, and you look at the interaction between both proteins, let's say A and B. In our case, what we did was to use those probes linked to the first epitope blocking monoclonal antibody towards synuclein. So because they're targeting the same epitope, we can only ever detect adjacent or aggregating molecules of synuclein with a maximum radius of 16 nanometers. And so at the end of the day, we end up with an assay that is specific to alpha-synuclein interactions. And if you get the right type of antibody, which in our hands has proved to be alpha-synuclein 211 in the case of human postmortem brain, or alpha-synuclein 46 in the case of in vitro applications, you can see that both these assays tend to be quite specific or enriched for the detection of oligomers. Now, once you have this interaction, what happens is you add oligonucleotide adapters and perform a ligation step, whereby only these oligonucleotide adapters can only be ligated if the antibodies are in close proximity. And finally, there's RCA polymerase that is can only use DNA that is in circular shape as a primer, and so produce a really big DNA bundle. So it's not to scale, obviously. The DNA bundle would be enormous as compared to this, and that allows you to then detect either by color or by fluorescence the resulting puncta. And so these tiny puncta that you can hopefully see on my screen here are the result of the assay by Brightfield. And so when this work was originally published, we were really shocked to see that in regions further apart from what we traditionally consider to be the war zone in PD, which is the brainstem and the medulla and the mesencephalon, such as the cingulate cortex, is a bit removed from disease, we saw quite a lot of heavy pathology as compared to controls, which was virtually undetectable by immunohistochemistry. And that's really interesting because at the end of the day, it also validated in a way the assay itself. Now, we had the tools to reveal the oligomeric pathology, but we also needed good models if we were to actually use these detection tools for therapeutic or disease modification through therapeutic compound testing. And so in the lab, a very talented PhD student, Steffi Janesek, in 2013 published this paper on our favorite transgenic animal, which is the alpha-synuclein overexpressing animal, or synuclein OVX for short. That is an animal that is a knockout for mouse synuclein, so it's a mouse non-background, and where we've packed the whole human synuclein locus and overexpressed it. So we have the synuclein OVX animals that express around 4 copies of the locus, or a fold increase of the locus, and then we have another control that expresses basically standard 1x, so to speak, of the locus. And what we could see was that there was a spatial temporal recapitulation of where synuclein really localizes in humans in the mouse, which was really comforting to see. But then also interestingly, 3-month-old animals don't seem to have any kind of problems, be it at the serology level or at the gait level. So we did a number of tests such as catwalk analysis or rotarod, and there were no deficits. But when we age the animals to 18 months, that's when we start to see neuronal loss in the OVX and deficits in the motor behavior. And that was really interesting because age is, at the end of the day, the greatest risk for Parkinson's disease. And so we thought, well, that's great. And it is actually one of the beauties of the model, that it's really nicely physiological in that way. The counterpart is that it takes a good 1.5 years or 2 to actually run an experiment. So it's what's great about the model and what's very slow about the model, let's say. And then another tool that we use a lot in the lab are iPSC-derived models. We use, in a standard way, cortical neuron and astrocyte monocultures, striatal monocultures, dopaminergic neuron astrocyte monocultures. I haven't put in here, but I should, co-cultures of microglia. Microglia we really only use in co-culture because we believe that that is the way to actually get them to express the most microglial-like morphology that they can. And then we also use organoids, even though today I won't be talking about organoids in these 2 particular stories. And so just to summarize, we have a number of assays and models that we can use to look at disease-modifying therapies for Parkinson's. And so we came into a collaboration between the labs of Erwan Bazar, Carlos Matute, and Richards at Oxford. And together with Gal Batan, who produced CLR01, we decided to evaluate its therapeutic benefit in our models and with our tools. And so CLR01 is a molecular tweezer. So the question that I often get in my talks is, what is a molecular tweezer? And so molecular tweezer is just a molecule that looks like a tweezer. It's nothing else. So when Gal, Frank, and Garrett actually produced these molecules, they thought it looked a little bit like a tweezer, and that's why it's called that and nothing else. And the way that it works, it actually just stabilizes very much like a tweezer. If you imagine the tweezer going on any particular bits, it will stabilize lysines. So if you look here in blue, we have the lysine being stabilized by the tweezer, in particular with the two phosphate groups depicted there in red. And right about now, a lot of people in the crowd will be thinking, well, actually, this must be tremendously toxic, because if it stabilizes lysines, at the end of the day, you will impair every biological process in the body associated to a protein that contains the lysines, which is pretty much everything. But the way this molecule actually works is that it's really, really low affinity. So it's really tremendously well tolerated, because the affinity is, its way of action is kiss and run. So it just touches the molecule and just becomes a nuisance for a second, a millisecond, whatever, a femtosecond, and then leaves. And it doesn't really impair any biological processes, and actually Gal has done extensive toxicology on it, but it really does, for some reason, create a nuisance for amyloid processes where there is a lot of overexpression or a really big abundant accumulation of something. There it does become a nuisance. And this is especially relevant to synuclein, because lysines are a lot of the crux of the matter when it comes to actually alpha-synuclein aggregation. And so we started the collaboration by getting some Lewy body extract from the lab in Bordeaux, from Erwan Bazar's lab, who produced the Lewy body extracts by ultracentrifugation of postmortem human material. And so they produced the Lewy body extract and the non-Lewy body associated fraction from postmortem brains. And so what we did with these two fractions was to just subject them to shaking and CLR01 and see what happened. And what we could see is that we didn't really get a lot of immunofluorescence or PLA signal from the non-Lewy body fraction, which wasn't truly a surprise, and there was no signal either, or not very much signal when we shook it with CLR01. But the Lewy body fraction did come up with some immunofluorescence and some PLA signal. And this was depleted or reduced when we treated them with CLR01. And anecdotally, we also put these preparations down the EM just in order to see whether we could see the same structures that they saw in Bordeaux. And indeed, we can see these roundish structures that they found in Bordeaux. And these are a little bit less present qualitatively when we treat the preps with CLR01. But more importantly, the lab in Bordeaux actually showed that these Lewy body fractions, or LB, were tremendously toxic in vivo. And so we decided to see whether this was also true in our dopaminergic iPSC-derived cultures. And what we could see off the bat was that Lewy body material is very cloudy. And actually, once we shake it with CLR01, it becomes less cloudy, which if you've ever actually prepared an aggregate, really tells you that about what's going on aggregation status. And you can see the deposition of just cloudy material on the cultures that has been cleared up by CLR01. But more interestingly, what we could see was that there was this really small increase in oligomers that seemed to be reduced. And more importantly, when we looked at process analysis here, we could see that axons, and I hope you can see this because it's quite tiny in the image, but axons labeled by TUSH1 here in the control were quite nicely preserved. But when we insult the cells with Lewy bodies, we get axonal blebbing or axonal degradation, whereby there's only where the axon used to be, there's little vesicles left and the axon degrades. So we can count this, we can count the blebbing, and we can count the process, the average process length of a culture automatically. And what we could see was that Lewy body material did seem to reduce the length and increase the blebbing, and thereby cause what we think is axonal degeneration. And we could rescue this by CLR01 treatment, so indicating that we could actually first insult and make the cells degenerate, and then also recover this phenotype by the molecular tweezer. In Carlos Matute's lab, a really talented PhD student, Paula, actually did the bottom two panels of this image, whereby she cultured neurons and astrocytes and looked at whether she could see cell death and in either or the neurons or the astrocytic cultures. And what she could see was that the Lewy body material seemed to affect neurons far more when looking at cell death than astrocytes. So there was more cell death in the cultures treated with Lewy bodies than in the cultures from astrocytes, which seemed to be actually resistant to the LB treatment. And then again, the CLR01 seemed to reduce this by its treatment. So this was really nice to see in the context of Lewy body material. But at the end of the day, there's many things in Lewy bodies and many aggregated things in Lewy bodies. So this takes us a little bit away from what it is that the core question, that is whether it's the early aggregation of alpha-synuclein or alpha-synuclein at all. So we decided to bring it back a little bit to a more defined insult. So we produced alpha-synuclein oligomers. And we also decided to use microfluidic chambers. And these chambers, what they do is they isolate neurons on either side of the chamber, and the axons can go through the grooves that you can see here, hopefully. But the somas remain either in the home chamber or the insult chamber. And by keeping the medium at a gradient, so higher in the home chamber and lower in the insult chamber, what we can do is to actually make sure that anything transported from the insult chamber back to the home chamber needs to go through active axonal transport. And so what we did was to insult the insult chamber and then also insult and treat with CLR01 and see what happened in the home chamber to see whether this would influence axonal transport at all. And indeed, we insulted the insult chamber. When we looked at the home chamber, we could see that from the insult chamber, we had gotten the red puncta that are PLA on the other side of the chamber, and that these were quite reduced when we looked at CLR01 treated cells, which really to us indicated that there was probably an interference of CLR01 with axonal transport of synuclein. Now, Revere 3 wasn't persuaded by this and wanted us to do a bit more work. And actually, we have also assays indicating that alpha-synuclein oligomers or alpha-synuclein monomers interact with dynein and kinesin, which we know from the literature happens, and that CLR01 prevents this. And that's how we got that extra data convinced in the end, Revere 3, that this was actually also the case in the microfluidic chambers. Now, because we had all this evidence in vitro, we decided to go in vivo and see whether this animal that I presented originally, the synuclein OVX, or the transgenic for human synuclein, also had phenotypes that were restored by the treatment of CLR01. And just initially, because at this point we hadn't published this data, we showed that wild-type animals don't really have a lot of GFAP activation in the span of the first year of life in the substantia nigra, as far as we've seen. However, OVX do start to have an enrichment in the GFAP stained area when they reach 12 months. And interestingly, when we look at microglial morphology, this happens earlier. So we don't see a difference in microglial number, which suggests that the activation isn't really a strong one. But we do see a difference in morphology from 6 months onwards. And this is really interesting because it follows the Ben-Barres hypothesis that the microglia activation comes first and then this is followed by the astrocytic one in time. But interestingly, this coincides very well when oligomers start to accumulate. So it almost seems like microglia are really the early responders to this type of insult in the nigra and the OVX. And it also gives us an instrument to look at aggregation in the nigra and see whether we can reverse the neuropathologic events because in a synuclein overexpressing animal, because we don't really have any Lewy body-like lesions in these animals. And so in order to test whether CLR01 could restore any phenotypes at 12 months in OVX, what we did was to deliver the drug subcutaneously for 2 months biweekly. No, sorry, 2 times a week. Biweekly is every 15 days. And so by doing this, what we looked at was at rotarod and catwalk analysis, so very minute, fine analysis of the gait. So that's what catwalk is, and rotarod, the ability of a mouse to just cope with a rod that is spinning with incremental speed. And what we could see was that there was a very small defect in rotarod that was corrected in the OVX animals that was corrected by CLR01 treatment, and that we could also see very early defects in the catwalk analysis in 4-foot swing speed, which is how the animal places, how long the animal takes to place the next paw on the catwalk, the speed of how quick it actually goes through the plate, and how many steps per second it does, or the cadence. And when we looked neuropathologically, it was these results really nicely mimicked what we were seeing in the behavior. So CLR01 was able to revert the oligomeric deposition of alpha-synuclein that we see in the OVX as compared to wild-type animals. And when we looked at the glial population surrounding it, we actually saw that astrocytes again were incremented, or the GFAP stained area was incremented in the substantia nigra when we look at OVX animals versus wild types, and that this was reverted by CLR01 treatment. Interestingly, this was not the case for microglia. So microglia are already quite amoeboid at this point, even in wild types. And what we could see was an increment in the amoeboid phenotype or morphology of the microglia. And so this was really interesting, or I found really interesting, because at the end of the day, it's a very coarse marker. You can't really know what a microglial cell is doing when they go amoeboid. In itself, it's nothing at all. But one would assume that if you treat any type of lesion or aggregate with synuclein, that there's going to be intermediate species released, and that someone has to clear that, and that it'll probably end up being the microglial cells. So it was really interesting to hypothesize that that's probably what they're doing. We also decided to use the state of the art or golden standard model from the Virginia Lee and Kelvin Look lab, that is the intracranial or intrastriatal injection of PFFs, or preformed fibrils of alpha-synuclein into the cranium, and use that as a model of Parkinson's. And as you can see, we get more lesions in the SNC than the VTA, which means that yes, we can actually do this and evoke pathology by phosphorylation of serine 129 and alpha-synuclein, and look at it in the right region of the brain. And again, CLR01 was able to downregulate the amount of pathology that we got in the SNC. And in parallel, in the lab in France, Amélie actually did really nice experiments using the original LB material that was that reduced the amount of neurons in the SNC stained by TH, as you can see quantified here. And this was rescued by the addition of the tweezer CLR01, as was the striatal OD. And she also looked at PK-resistant alpha-synuclein and could see a reduction of that, as well as the area stained by the antibodies in F1. So hopefully I've left you with the following conclusions of this first part. The CLR01 is able to revert the toxic effects caused by LB extracts in iPSC-derived dopaminergic cultures. That CLR01 is able to correct age-related neuropathology and motor defects in synuclein OVXs, and that it is able to rescue the neuropathologic effects of intracranial injection of mouse PFFs and Lewy body extracts. And Jonolano and Patri are now continuing this type of work in the lab and are using newer molecules that we think are really beneficial and that can serve as disease-modifying therapies, both in the in vitro and in the in vivo models moving forward. And then the other short story that I wanted to tell you about is REST is a neuroprotective factor for PD. And since hopefully I've already convinced you that there is a synuclein oligomeric accumulation in synuclein overexpressing animals, I also wanted to add that before we had the synuclein oligomer assay going, or the ASPLA, the PLA of the oligomers, we didn't really, the original paper is called a synuclein overexpressing animal because we couldn't find Lewy body-like pathology. We couldn't find phosphorylation of SYN129. We couldn't find anything that looked like a fibril in these animals. And so the paper ended up calling itself overexpression of synuclein because we thought that that was it. There was no aggregation of synuclein, just an overexpression, just buckets of synuclein versus of monomer. But now at this point, like we had already established that there was aggregation going on. And so we also had performed way back when proteomic analysis, which revealed dysfunction of several metabolic pathways, many of which are really or have been really implicated throughout the literature in Parkinson's disease, such as, perhaps this is a little bit tiny, but here it says oxidative phosphorylation mitochondrial dysfunction. And Brent, the co-author of this paper, did this proteomic analysis and then went back into the lab and confirmed this mitochondrial dysfunction. And the mitochondrial dysfunction was confirmed by many different bits. So basically, a lot of Western blotting was performed in order to see that all the markers that were dysregulated indeed were dysregulated. We looked at complex 1 function, which was reduced in the midbrain. ROS was increased in the midbrain. And so we decided that this was great, but it was still not unexpected, and it was still not very revealing for a new target or anything like that. And at the end, we're really interested in therapeutics, new therapeutic targets, etc. So what Brent very cleverly did was to actually look at that those hits of the proteomics and look at master regulators of that network of hits that he got. And one was synuclein, which made sense because it's the overexpression, the protein that is overexpressed in that model. But another one was REST. And REST had been found to be really important in healthy aging by the younger group. And so we decided to look a little bit into this protein. And the first thing that we did was to actually look at wild-type and synuclein OVX animals in young and aged animals. And what we saw was that there was no nuclear REST, and REST is the transcriptional repressor. So if it's not in the nucleus, it's not active because it needs to repress genes that obviously are in the nucleus. And so when we looked at the SNC of either wild-type or OVX animals, we couldn't really see anything in the SNC. There was basically no levels of it. However, when we looked in the SNR, which is the substantia nigra pars reticulata, which is protected in Parkinson's versus the SNC, which is full of GABAergic neurons that can also be labeled with NEFH. We saw that not only there was REST in their nuclei, but they could upregulate it in the transgenic animal. And not only that, when we look at oligomeric deposition in these animals, we can see an increased oligomeric deposition in the dopaminergic neurons, but not in those of the SNR. So it was really interesting to see this associated phenotype of potentially a rescue and potentially protected cell type and a potentially vulnerable cell type, showing REST and either rest and less oligomers or no rest and more oligomers. And so because Brent had found this widespread mitochondrial dysfunction, we also tried to look at the mitochondrial network. And so it's very difficult to do this in tissue. So already at 12 months, it's quite complicated to see. But if we concentrate on the 3-month animals, what we can see there is that the aspect ratio or the elongation of the mitochondria is worse always in the SNC when compared to the SNR. And that the number of objects or mitochondria per cell is increased, meaning that those mitochondria are fragmented. And they're even more fragmented in the transgenic animal. And that seems to be also the case in the 12-month cohort, but there the mitochondria are already quite at the limit. So we can't really analyze the delta that we have to measure is very poor. But still, this correlated again with the mitochondrial picture that we had, the oligomeric and the presence and absence of rest. So we decided to go in vitro to be able to functionally manipulate rest and see whether we could see associated benefit for mitochondria or not. And what I forgot to put in here is that we have a reduction both at the protein level and at the mRNA level of rest in these cells, but you can find the figure in the paper if you're not persuaded to believe me. And what we saw was that when we knock down rest, we can reduce the aspect ratio, again, the elongation of the mitochondria, and increase the number of them, meaning that they're more fragmented. And so that rest knockdown is not particularly good for mitochondrial health. We also looked at cells undergoing mitophagy in both the whole population and also selectively in the cells that have been transfected by the guides, by the CRISPR guides for the rest knockdown. And we could see that indeed there was less mitophagy in the cells that were knocked down for rest. And we also looked at the mitochondrial membrane potential and the ROS associated to mitochondria. And we could see a reduced mitochondrial potential, indicating that there's probably some sort of problems with the respiratory chain in order to maintain the potential, and an increase in ROS, again, associated with mitochondrial dysfunction, meaning that rest knockdown reduces mitochondrial health. So in light of this, we decided to do the counter experiment or the overexpression. And so we used the plasmid to overexpress rest in the same cell type, the SHSY5Y system, which is really amenable to transfection and genetic manipulation. And we insulted these cells with MPP+. And obviously, when you insult SHs with MPP+, a number of them die, as illustrated here. And in cells overexpressing rest, this difference no longer was significant. But more importantly, when we looked at the mitochondrial networks of starved cells, which is how we get them into a Parkinson's-like state, we can see that when we overexpress rest, we can increase that aspect ratio or elongate that mitochondria back to basal levels. And once we did these experiments, we were really perplexed by the ability of rest to influence mitochondrial morphology. And therefore, we did have a number of antibodies in the lab, amongst them PGC1 alpha, NRF2, PPAR gamma, etc., that we could test to see whether any of these were a hit for rest overexpression. Because at the end of the day, you probably have a mitochondrial master regulator being touched, let's say, by rest. If you're seeing all these effects on mitochondrial morphology. And neither PPAR gamma nor NRF2 were upregulated by rest overexpression, but PGC1 alpha was. And so again, we went to the other cells, to the knockdown cells, and we saw that indeed when we knock down rest, we can actually get a downregulation of PGC1 alpha. And that was quite interesting because we also had another antibody that worked for staying for PGC1 alpha. So we were able to go back to the mouse brain and actually interrogate whether the different neuronal populations that we originally were describing, the SNC vulnerable SNC and protected SNR had PEGC1 alpha nuclear levels. And what we could see was that in the SNC, there's barely any PGC1 alpha, but in the SNR, we can see, labeled by NEFH here, sorry, instead of SNR, we can see that not only are the levels, the basal levels, quite high in the wild type animals, but that this is increased in the synuclein overexpressing animals, which ties in together all the story and was actually quite a nice way to link the mitochondrial health back to rest. And by this point, we wanted to publish this story, but obviously someone else had gone and published something also very interesting, which was the Kawamura et al. Paper, which I think was published in 2019. And what they very nicely showed in their paper is that rest is sequestered away from the nucleus in Parkinson's disease to Lewy bodies. And when that paper came out, we thought, well, we don't really have Lewy bodies in this animal because we've looked, we never found anything of the sort. As they say, no PSIN 129, no fibrils, no higher order aggregates. And so we thought, well, but we do have oligomeric accumulation beyond 12 months and beyond. So why don't we use conventional PLA to look at the interaction between rest and synuclein and ask whether even earlier aggregates, because the Kawamura paper hypothesized or poses that at the end of the day, it is the lesion that sequesters it away from the nucleus, and that's how the neuron ends up not being able to cope with the additional stress. What if early aggregates could also play this role? And maybe the interaction between synuclein and rest is increased in the cytosol in aged animals. And indeed, when we went and did this experiment, that was the case. There's very little interaction between rest and alpha-synuclein in wild types and synuclein OVXs when they're young, but when they age, this remains the same for the wild types. But the interaction is increased in the OVX animals. And so hopefully I've left you with the idea that rest protects neurons from mitochondrial and alpha-synuclein stress via PGC1 alpha, and that this neuroprotective mechanism is lost in neurons and dopaminergic neurons, rendering them vulnerable to disease, particularly Parkinson's. And now it is for Dalila Ciceri and Begum Kurt to actually translate these findings in vivo, and that's what Michael J. Fox has given us funds to do, in order to assess whether rest is a therapeutic target for PD in modifying rest functionally in vivo through viral injection. And we're going to perform these experiments in both the transgenics that we already know and that we have built this story on, and in the PFF injection model from the Virginia Lee and Calvin Look lab that I previously mentioned through the PFF intracranial model. And what remains is to thank all the people involved in all this work. Firstly, the past people, Fabio, Paula, Carlos from the group in Spain, way back when we did the CLR01 story, Thomas, Frank, and Gail, who actually made CLR01 happen to begin with. So we're instrumental in that story, as well as the group in Bordeaux, Emily, Jaman, and Erwan. The people at Oxford who bared with me while I learned electrophysiology and neuropathology with them, Bradley, Steph, Pete, Javier, and Laura. The team at BMS who gave me my first shot at a grant, Reach, Shamik, Richard, and the rest of the BMS team, as well as Liam. Of course, Richard, for giving me the opportunity to work in his lab for that long, and Jane and Sally, who were instrumental for the IPSCs. And my team that has now expanded into this present beautiful team, part of them at Oxford, Ajantha, Quinn, Faye, and Naroa, and part at Achucarro, Patri, Beg, Alba, Lierni, Dalila, and Yon, who are amazing and whom without I couldn't really do any of this work. And finally, to all the funders who still believe that, you know, it's important to fund young scientists in new labs as well. Thank you for your attention. Thank you very much.
All right, thank you very much, Nora, for a very interesting talk. So the paper is now open for discussion. As I said earlier, if anyone is interested in making a questions to Nora, please do so by using the raise hand button on the top of your screen, or by sending me a message through the chat stating your name and affiliation and your interest in making a question. There is no need for you to write down the question, since I will give you the floor to pose the question yourself. Okay, so while we wait for the first questions, let me remind everyone that the CIBERNED Webinar Series are held every other week on Thursdays at 3:30 p.m. The program for the upcoming few months is already posted at the CIBERNED website. So if you're interested in looking at the program, please go there and check every webinar from now until the end of June. Also, if anyone is interested or know anyone in her group or other groups of CIBERNED interested in participating, presenting data, etc., please just send me a message or to Teresa Iglesias, who is the training coordinator in CIBERNED, and I will try to accommodate in any free slot that can be available in the next few months. Okay, so time for questions then. I don't see any raised hand yet. No messages coming through the chat, neither. All right, so we have the first question coming from Carlos Matute. Carlos, please go ahead.
Hi, everybody. Nora, congratulations for the work. Very, very nice. It raises many questions, so I have made them before the seminar to you, but they came to my mind right now while listening to you. So one of the pathways which was touched in your pathway exploration was, I think it was the second one, glutamate degradation pathway. So could you please speculate or tell us what is the meaning of this, or how you interpret this alteration in this pathway?
Well, glutamate always obviously rings a bell for astrocytes, but I have to say we haven't really explored anything beyond the mitochondrial defects at the metabolomic level, which I think would be very interesting. And the other thing that we haven't really explored, but I'm actually dying to do, which is tangentially associated, is lipids. Because obviously if we have all this metabolic dysregulation, I expect that the lipids are also going to be dysregulated. And as you probably already know as well, there's a lot of dysregulation happening in the nigra for lipids that we really don't quite fully understand, but we think it also could impact not only signaling, membranes, trafficking, all sorts of things that are, again, intimately linked to the homeostasis of the dopaminergic neuron, particularly when they have such intrinsic arbors that require such heavy ATP demands, and that's again why we think that they're so mitochondrially dependent. But yeah, I haven't elaborated any more on the glutamate axis, apart from speculating that it might just come from the astrocytic component. I can't really tell you much more.
All right, thank you, Nora. Carlos, more questions?
I have a second question.
All right, go ahead.
So it's about the mitochondrial defects assessment. So did you do metabolic assays? So in other words, did you measure oxygen consumption or these kind of parameters which are typical, or you just rely on morphological and biochemical data exclusively?
Yes, that's a great question. So we tried a lot of seahorse. So a part of the project that I haven't really presented is that we also looked at the alpha-synuclein triplication patients, which are the homologous and the iPSC models for dopaminergic neurons in a human flavor, so to speak. So they have mitochondrial defects. They have mitochondrial seahorse dysfunction, basically the whole curve is shifted down so that all of the things that you can think about are basically a little bit reduced, a little bit not as functional as control neurons when we differentiate them into neurons. They have oligomers, and they're basically the sister human model to the animals, and they also have a rest impairment, interestingly. So we spend a lot of time trying to overexpress or knock down rest through viruses at that point, and we could never get the sweet spot. And if anyone, please email me if you know, because the thing is that what we always found was that the transduction itself, both of the control virus and the rest-associated virus, completely changed the mitochondrial respiration landscape of the cell to something that was nothing like the control one. So we never could find the ideal way of getting rest into the iPSC to actually address that question, which would have been great, obviously. And we didn't try to put any slices in the seahorse, which I think now is something that they can do. But at the time when we did this, I don't think that there was so much literature or trials on actually putting slices into the seahorse. But yeah, it is something really interesting that we might actually do in this continuation project.
Thank you very much, and congratulations for the work.
Thank you.
All right, thank you, Carlos and Nora. Any other question from the audience? Okay, I don't see any raised hands. I don't see any message coming through the chat. Okay, so this is the last call for questions to Nora. If anyone is still want to make a question. No?
You can always email me if there's anything that pops into your head later on.
All right. Okay, so if there are no more questions now, then this is time to close the session, but not before thanking Nora again for a great talk, and also for her willingness to share with us her work. Nora, thank you very much. Welcome to CIBERNED community. And I think, I think we're gonna close here because there are no other questions coming. So thank you everyone for attending. Thank you very much, Nora. And I wish you all a great afternoon. So goodbye.
Thank you. Bye.
Bye.